Hi, I'm Brooke DeVries, and today we will be talking about the basic structure of DNA. Now, DNA is a nucleic acid, which is one of the four big molecules. The others are lipids, carbohydrates, and proteins. The monomer of a nucleic acid is a nucleotide. So here we have a nucleotide. There are three pieces to a nucleotide: the phosphate group, 5-carbon sugar, and a nitrogen base. 5-carbon sugar is thus named because it has, well, 5 carbons. Starting at the oxygen, we name them in a clockwise direction: 1', 2', 3', 4', and 5'. This will be important in the structure of DNA. Now, there are 4 nitrogen bases in DNA. This one happens to be cytosine, and we'll talk about what that means here in a minute. The 4 bases are adenine, thymine, cytosine, and guanine, again, in DNA. Pyrimidines are thymine and cytosine, again, this is cytosine. Now, purines are adenine and guanine. Chargaff's rule states that pyrimidines must bond with pyrimines. More specifically, adenine must bond with thymine, and guanine must bond with cytosine. Now, this is one nucleotide. It's not going to do much, so you have to have a whole chain to make up DNA molecules. To make this chain, the 5-carbon sugars bond to one another at the 3' and 5' ends. So the 5' of the next will bond to the 3' here, 3' would bond to this 5', making a chain. These would go in the middle of what's called a double helix structure. Double helix is sort of like a corkscrew shape. Looks something like this. Now you'll have your bases here. Again, adenine will bond with thymine, and guanine will bond with cytosine. Now, this strand is called the backbone of the DNA, and that's composed of the phosphate group and the 5-carbon sugars. Now, adenine and thymine, due to their structure, have 2 hydrogen bonds between them, while guanine and cytosine have 3. This will be important in DNA replication. Now, the 2 backbones of the DNA are what's called anti-parallel, which means they're parallel, so they won't meet, but they're running in opposite directions. So let's say this strand runs from the 5' here to the 3', and this one runs from 3' to 5'. Again, this will be important in DNA replication. One strand will be what's known as the leading strand, while the other is the lagging strand. And this is the basic structure of a DNA molecule.